Good morning, everyone. It's a pleasure to be here. My name is Bruno Contreras. I will be presenting you today a few tools that we have developed over recent years for the analysis of pangenomes. I've put the link to this presentation in the chat, and you can also scan the QR code on the screen if you want to get a copy of the slides because there are links on them, and it might be useful for you if you miss something from the talk. Okay, so first of all, I will present myself a little bit. I'm working at the Estación Experimental de Euladei, an institute of CSIC in Zaragoza, in Aragon, which is devoted mostly to agriculture. We have departments that work on soil and irrigation, departments that work on plant biology, and a department that works on genetics and breeding. And the work of my lab is, as you can see in the logos on the screen, is part of the Conexión CSIC and also part of the Spanish node of Elixir. So I'm going to start by showing you some basics about pangenomes, okay? In this slide, you see a couple of figures that I took from the paper that we thought we still think is the main paper, the pioneer paper in bringing the ideas that gave rise to pangenomes. Was this paper by Welch in 2002, where the authors for the first time compared the genomes of three strains, in this case of the Escherichia coli microbe. Okay, so you can see in this Venn plot here there are three strains. One is non-pathogenic, which is the K12 strain, and then another one is uropathogenic, and the one on the bottom is enterohemorrhagic. Okay, so these three strains of the same species of microbe, when they compared their genomes, they realized that some genes were found in all of them, in the three of them, and they are marked in the horizontal bar below. When you see here the core collinear orthologous genes, these are genes in this example here, like cell C. You see the gene takes exactly the same place in the genome on top and the genome on bottom. Okay, so these are collinear. They conserve the position, and they are present in both genomes. But they also found for the first time that there were some genes that were unique to some strains, not found in others, and these were called the accessory genes, which are in some cases strain specific. It could be specific to one strain or to a group of strains, and these kind of genes are what represent the presence of absence variation. Okay. 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 So a few years later, there was another landmark paper on a different bacterial species, now by Tetelin and collaborators. In this case, they studied a larger number of genomes, and because they had more genomes, they were able to see for the first time a little bit about the dynamics of the genome in terms of growth of number of genes and how many of those genes are shared among strains of the same species. Okay, and these two plots that you see here are what I call the core plot and the pan plot. The one below, as you can see, the number of genes added by a new strain that you analyze of your species of interest. Every new species you add to the pool, the number of new genes gets reduced. Okay, and that's because there is a convergence. There is a point where it doesn't matter how many genomes you have, you will not find any new genes. Okay, and the top one plot is a different way of the same problem. Is how large is the total number of genes when you keep adding genomes. Okay, in this paper, they have 8, 9 genomes, and they realized that there was a trend. It seemed in this case that you would expect some more new genomes when you if you manage to sequence and annotate a new genome, you would expect some more new genes to appear. And this is what they called an open pangenome, as opposed to a closed pangenome. So open pangenomes are those that you, according to the evidence you have, which is a number of genomes that you've studied, you expect that it will keep growing. So that's an open pangenome. But there are others. This is specific to the domain that you're looking at. There are others that are closed, so that they seem to reach a plateau. And after a few genomes, you won't see more genomes coming into the pool. Okay, so these two papers prompted a lot of interest, and new names and new concepts were needed to understand what was going on. Okay, so in this slide, what I'm trying to do is explain some of these nomenclature and concepts. Okay, so on the left side up, you see a Venn diagram of three toy genomes, the genome A, B, and C. And the numbers that you see are genes. Okay, so in this example, there are 101 genes which form the core genome because they are found in all three genomes. But you can see other numbers. For instance, there are 22 genomes that are unique to genome C. Okay, the bar plot on the right is like a summary of this Venn plot. You see 3 bars with different colors. The white bar is what we call the core gene, the core genome for this example. So has a height of 101 because there are 101 genes shared among the 3 strains or genomes of interest. There are 44 genes which are found in 2, but not in the 3 genomes, and there are 82 which are unique, which are in this case found in only 1 genome. Okay, these 2 classes together, the red and the orange, the cloud and the shell together, is what I like to call the accessory genome, as opposed to another term which is quite frequent also in the literature, which is called the dispensable genome. And so you'll see it in both ways. I prefer it in the first way, accessory, because we don't really know in many cases whether it is dispensable or not. Okay, but you need to know that this is also valid nomenclature. Many people use it. And you also see in the table below that the cloud genes, which are red bar, is the red bar in the plot. You can also find it in the papers as private or unique genes. Okay, and the table below also shows you an interesting outcome of this kind of analysis is that you can see in each arrow you see genes which you see a set of genes which are shared in 1 or more genomes. Okay, so the second row, for instance, has gene 2A from genome A, genome 42B from genome B, and so on. Okay, so you can also see that genome A and B for that particular example have only 1 copy, but genome C for that copy has 2 genes. Okay, so this happens in reality, and so you have to deal with it. Sometimes the genes that play the same role in the genome have a variation in the number of copies, and these genes we call in paralogs. Okay. With Pablo Vinuesa, you see him in the picture here. We discovered many years ago when I was a postdoc in Mexico that if you try to summarize at the functional level, broad functional classes, the different compartments of pangenomes, you see that they're not all the same. It seems pretty obvious. In this example, on the left, you have 13 gamma proteobacteria. So a lot of bacteria from a very broad taxonomic class. And on the right side, you have 20 different strains of Escherichia coli. In this case, they are very close genomes. But regardless of the taxonomic or evolutionary distance between the material that you're looking at, it was pretty obvious at the time and still is that when you look at the functions of the genes that take part of each compartment, they're not the same. And in this case, the colors in the bars represent very broad functional classes from an old classification called the COX. And you can see that some of the colors are shrinked in some classes and enlarged on others. Okay, and well, that's basically the main message from this plot. And this is the same. Over the years, we discovered that this picture is also found when you look at plant genomes. So this is not a property only of microbial genomes when you do this kind of analysis. When we did it some years ago with barley and Arabidopsis thaliana, we found a similar picture that some genes, some classes of genes you tend to find mostly in the core, and some others you tend to find mostly in the shell, for instance, okay? And in the case of plants, it's interesting to see that the gene families related to defense against diseases and against pathogens are in many cases found in the shell, which tells you that the evolution has prepared plants to combat diseases and stress sources which are specific to the place they live on. And you won't find the same genes if you move to a different geographic area. And finally, in this slide, I wanted to show you that the last step in the development of this pangenome concept has been the pangenome graphs, which have been around for some years already. Also started in bacteria, but they're gaining a lot of momentum now in the human genomics community because they are being used to summarize collections of human genomes, and that's called the human pangenome. And they are often quite hard to represent graphically. It's quite hard to explain to someone what a pangenome looks like because they are very complicated structures to paint. I like the tube map that you see below on this plot because I think it's easy to understand if you put it that way. But that only works if you look at a particular region in the genome. If you try to do that with a whole chromosome, is really a mess. Okay. And in these final slides of the general concepts, I just wanted to recapitulate the history of pangenomes. And you can see that the paper I mentioned in my second slide, that the telling paper, according to this review, was the first paper about pangenomes in 2005. Well, I could argue the Welsh paper probably in 2002 was probably the first one, but that's fine. You see here some landmark studies, first on bacteria, then soon you'll start seeing, for instance, in 2007, these ideas were already discussed in plant, in particular in maize. Also, although it wasn't fully developed, it was more like an exploratory study. In 2009, there was already a human pangenome built using 3 genomes. And then as years passed, the dimension of the studies kept growing. Okay, so for instance, in 2018, there was already pangenome of more than 1,000 yeast isolates. And in 2019, there was already a human pangenome of more than 900 individuals. In this timeline, I show here the 3 software tools that we developed over the years, more or less at the time they appeared. And So the first one is get homologs that was developed with bacterial studies in mind. Second one was get homologs EST, which was developed when I started applying these ideas on plant data. And many of the data sets we had were low quality transcriptome sets and mixed with genes annotated from genomes of different quality. And later, we worked on the get pangenes pipeline, which already uses high quality genomes. Okay. Okay, so we are already half time, more or less 15 minutes. I'm going to now spend the rest of the talk, the remaining 15 minutes, presenting what applications these three tools have, okay? And for that, you need to, in each case, you need to think about the problem you have at hand and which is the right algorithm for you. So this table is a very broad summary of the three tools. With the main features, okay? So in the first row, you see get homologs, which is the oldest algorithm that would take as primary input, generally protein sequences extracted from gene bank files. So which is what you usually do when you're working with bacteria. So if you have 10 gene bank files for 10 bacterial genomes, you can use them as input for get homologs. The primary engine to compare sequences in this case is going to be BLASTP, or you can also try Diamond if you want to do very large data sets. Alignment coverage, which is a key parameter in the algorithm, in this case is computed with respect to the query sequence. So if you're comparing genome A against genome B, then the coverage would be computed on the sequence A, okay? Then when you do it the other way around, B against A, sorry, then you compute the coverage with respect to B. These are fine detail parameters which are important. The clustering algorithms that are integrated into get homologs are ortho-MCL, coq triangles, and also the classical bidirectional, the BLAST bidirectional best hits, okay? And in this case, there is no filtering of isoforms because usually isoforms at that time when we were developing these was not a problem in bacterial annotation. So each gene would have typically only one isoform. The second row is the get homologs EST algorithm, which takes as input now instead of proteins, it takes nucleotides. Okay, so this is already a big difference. While get homologs can be used to compare bacteria that have a certain evolutionary distance among them, you cannot do that with get homologs EST because you use as input nucleotides. And because nucleotides are less conserved, the moment you get far away in evolutionary time, then there is no more signal to compare the sequences, okay? So get homologs EST has been used mostly to compare genotypes, cultivars of the same species, and sometimes maybe an outgroup. But the short message is that you need to have materials which are related, okay? The primary engine in this case is going to be BLASTN, and the alignment coverage is computed with respect to the shortest sequence. And the reason for that is that way you can mix high quality annotated sequences with poor quality transcripts, okay? The clustering algorithms supported are ortho-MCL and bidirectional base hits. And in this case, there is an isoform filtering step, which is very important, which means that if you have several isoforms of the same quantitatively expressed from the same locus, you summarize them. You don't use them all because that will confuse the clustering, okay? And finally, the most recent algorithm, which is called get pangenes, and we have applied it mostly for plants so far, takes as input genome files in FASTA format plus annotation files in GFF format, okay? So in this case, you assume that the quality of the assemblies is good enough. And then for the first time, we are using synteny or collinearity explicitly from the beginning. The primary engine is going to be Minimap2, and the coverage is computed from the alignments produced by Minimap2 whole genome alignments, okay? And can also be used, and this is an interesting feature of get pangenomes. This approach allows you to confirm or reject presence-absence variations. Because even if a gene is not annotated in a genome, you can still tell whether a region in a genome is actually part of a cluster of genes, which is what we call a pangene, okay? And this is a common problem in plants because often genes are missed in the annotation, perhaps due to lack of expression data in a particular tissue. So you don't see gene expressed, but that doesn't mean necessarily that the gene is not in the genome, okay? You can later confirm whether that gene is still there or not by comparing to the others. The cluster algorithm in this case is a greedy algorithm which is comparable to the bidirectional best hit. And in this case, isoforms are not filtered because the computation is done at the genome level. So when you actually get to the step of producing clusters of genes, all isoforms are taken. So here you have flowcharts of get homologs on the left and get homologs EST on the right. There's a lot of things going on here. What I want you to look at is mostly at the boxes in the bottom, which is the output of the algorithm. You can get a few things out of the algorithms. You can get clusters. This is the main output of these algorithms. Sequence clusters, which are in FASTA format. They can be both amino acid and nucleotide clusters. You can also get estimates of the core and the pangenome as it grows when you add more individuals to the pool. You can get matrices of average nucleotide identity or matrices of percentage of conserved clusters. And you can also get pangenome matrices, which are these matrices as the one I showed you before in the toy example that summarize presence, absence, and number of copies in rows, okay? Okay, I'm gonna move to the next slide. Get pangenes is a slightly different approach. In this case, there are three main steps. I'm not going to explain it in a lot of detail. I want you to the take-home message here is that the kind of output that you get is virtually the same as we had earlier. Okay, although the computation is completely different. At the end of the process, what you get are sequence clusters in different format. They are FASTA files. You can also get estimates of the size of the pan and the core set, and also these matrices where these pangene matrices, and also Annie and box matrices, okay? So I have roughly 6 minutes left. How do you install this software? Okay, so in this table, may I remind you that the link to these packages and to these slides is in the chat. So if you open the slides yourself later, you'll see that these links here take you to the installation instructions of the three packages that we're talking about today. Usually I install them from the GitHub directly. I would clone and run an installation script, but you can also install it from Conda in all three cases. In fact, um, when you install get homologs, you get homologs EST at the same time or the other way around. They are installed together. Get pangenes is separate. And the first two options are also available as Docker containers, which can also be converted to Aptainer if you need. And they are bundled with another analysis tool, which is called Get PhenoMarkers, that I'm not going to talk about much today. but it's handy because you can use straight away the clusters you produce with get homologs. You can use them to get very nice, robust phylogenies of the data that you are analyzing. The downside of using the Docker is that there is no support for high performance computing, uh, uh, which instead there is when you use GitHub or Conda. Okay, so if you are going to analyze large data sets with get homologs, it's better to use the Conda or the GitHub version. So where is the documentation for the software? In this table, you have links to the manuals, the tutorials, and even to a terminal demo that we created a few months ago for pangenes in particular. So there is a lot of documentation backing up this software. In particular, Pablo Vinuesa, my colleague and co-author of Get Homologs, and me have been updating the manuals of Get Homologs for years. And I think that's one of the reasons that the community have been using this software a lot. And so the manual and the tutorials will show you how to start from scratch and how to analyze the data, okay? These final slides are only to summarize what these programs are about, okay? So here you have a graphical summary of what I think, that's my view of the situation. When people talk about several genomes of the same species, mentally, I think we all have an idea that the A, B, and the N genomes are going to look like that. So individuals on the same species, their genomes are so similar that you expect them to have the genes conserved in all of them. The tools that I have been describing today will help you realize that the situation is more like the one you have on the right side. So there is clearly variation both in prokaryotes and in eukaryotes, the level of gene content, so that individuals of the same species or related species share some genes, but not others. And there's also variation on the way these genes are mapped in the genome. So here in the example, you have, for instance, a gene that is flipped because there was an inversion of on that part of the chromosome. And sometimes those events, those structural rearrangements have an effect in the function. Sometimes they don't, but sometimes they do. And we have examples of that in plants. And I want to terminate by telling you that here in the second row, you have on the right side the image that I imagine we all have when we think about a gene that is shared among individuals of the same species. We think they're going to be sequences that are roughly the same length, that they can be aligned easily. But using the tools that I have described, you frequently find out that the situation is not that nice, is that genes of the same locus, a pangene, that's the word I would use, are frequently different, particularly when we have been looking at this in rice recently, using Asian rice, using very high quality genomes. We found that it's very common that there is polymorphism in the length of the genes, and there's very common differences at the UTR level, but also at the exon level and at the intron level. Okay, so what is coming up, the picture that is emerging from this kind of analysis is that there is a richness of variation at the level of gene content in genomes, okay? And I hope that if you're interested in these questions, I hope you would try out some of these softwares, and I hope they will be useful for you in your projects, okay? And this is just the goodbye slide. This is the institute we are working on. And that link there is the blog where we regularly post questions about computational biology, not necessarily our own software. We write about lots of things, and I hope it will be interesting for you.